Welcome to the PharmaSource podcast.
Cell and gene therapies are moving fast, but the manufacturing infrastructure needed to support them is struggling to keep up. Induced pluripotent stem cell-based therapies represent an exciting frontier in medicine, yet turning a promising lab process into a scalable clinical-grade product remains one of the industry's toughest unsolved problems. Today's guest has spent over 2 decades working at every stage of that journey, from early stem cell research at the Roslin Institute, through clinical roles at Novartis, to setting up a first-in-human trial for an iPSC-based cancer vaccine. Sullivan is now founder of Linville Bio, where he advises biotech companies on how to navigate the manufacturing and regulatory complexity of getting iPSC therapies to clinic. In today's conversation, Stephen shares what makes advanced therapy medicinal product manufacturing fundamentally different, why companies consistently underestimate the CMC challenge, and what CDMO selection actually looks like in this space, and where the biggest risks tend to catch companies off guard. Stephen, thank you for joining me.
Pleasure to be here, Dora. Thanks for the invitation.
Can you just start by telling us a bit about your background? I know it's really exciting, and how you ended up where you are today.
Well, I suppose I originally trained as a developmental biologist, and particularly in 1997, it was a huge year. Dolly was cloned, which meant that you could reset a mammalian differentiated somatic nucleus, so you could make a cell forget what it was. And then human pluripotent stem cells were derived for the first time. So you could start to think about artificially wiping a cell and then scaling it and then differentiating it into different cell types. So as you said, I started off some of my early career in the Roslin Institute, which is associated with cloning and stem cell biology. And very much my early work focuses on how to make a cell forget what it was and then to make it into different cell types, and that all naturally led to an interest in induced pluripotent stem cells when they arrived on the scene, and really their remarkable capacity to be a potential new manufacturing unit for a whole plethora of different cell therapies. So over time, my focus shifted away from the basic biology towards the translation of stem cell science into therapies, and that led to roles, in involving clinical translation and manufacturing strategy, including some time at Novartis. More recently, as you say, I was involved in setting up a clinical trial for an iPSC-based cancer vaccine. And today I run a consultancy company which helps biotech companies and other companies either enter or navigate this kind of new emerging area of induced pluripotent stem cell therapies and get them to the clinic, because we can see a lot of clinical trials in other emerging modalities, but it's really about working together effectively so that we can get more therapeutic products into the clinic and then ultimately, across the globe with maximized clinical impact.
For anyone who isn't familiar, can you explain what iPSC therapies are, where the market's heading?
Induced pluripotent stem cell therapies, or iPS cell therapies, we spent 20 years now refining and understanding the basic biology around those cells, and essentially these cells have 2 properties that are very, very important. One is that they have the capacity to self-renew, so you can take a small number of stem cells and grow them up in bioreactors to the cell number that you require, and you can almost indefinitely culture the cells up in number. And then the second thing is they have this capacity called pluripotency, which means that you can direct their differentiation into different cell types. So the progeny of our stem cells can be used, like for example, to treat cardiac conditions or neurodegeneration or diabetes or a whole range of different diseases, producing cells that can mitigate the pathology and deal with the underlying injury, or in that case. So I think the key thing is that iPSCs in particular allow us to create a renewable manufacturing platform for therapeutic cells. And that's where the real excitement around it is. And in many ways, iPSCs represent one of the first truly scalable models for regenerative medicine.
Why is that?
Normally, you have the problem of the more material you remove from the patient, the worse they get. Here, you can take stem cells, grow them in number outside the body, so it's scalable, and then you can differentiate into different cell types. So you got a whole plethora of different indications that could potentially be treated with different cell types. And you don't even have to think about differentiating the cells into just one cell type. You might be differentiating them into a progenitor population, which then introduced into the patient will respond to the niche in an appropriate manner. And then that would kind of solve your problem, be it pathology or injury.
So you've been in this space since really at the start of its inception, 25, 30 years ago. How has the manufacturing challenge changed over that time? We've obviously spent a lot of this time understanding the basic biology. Where's the manufacturing going?
Yeah, I would say, if you're listening to this podcast and your world is established modalities, things like antibodies or, small molecule therapeutics, it's a kind of different world when it comes to cell therapy and iPS cell therapies in particular. We spent the last 20 years really just understanding the basic biology of the cells, and researchers were kind of asking fundamental questions about how to reprogram cells, how to maintain this pluripotent ability, this ability to make different cell types, and how to direct differentiation into particular cell types. And that work was essential, and it's enabled the field to move forward to where it is today. But today we're seeing a very different phase, which is moving away from the proof of concept biology towards basically tackling the challenges that face industrialization of therapeutic development. So the real challenge now isn't whether we can generate different cell types that we need. We can. And the challenge is whether we can manufacture these cells reproducibly, safely, and at scale under GMP conditions. And, we're dealing with, like, for example, living cells, right? So, if you start with a drug you can irradiate at the end of your manufacturing process, that's okay. You've got your sterile therapeutic product, and then you can put that safely into your patient. Here, when we're dealing with living cells, irradiation, you can't irradiate your cells because that'll kill your cells. So you have to start in fully kind of sterile conditions and maintain those all the way through. So there's a degree of complexity over and above standard GMP manufacturing. And, the reins of translation are firmly in the hands of early developers. And these early developers might have limited understanding about the decisions that they have to take and the downstream consequences of those decisions on the manufacturability of their therapeutic product. So frequently we can find early developers that make decisions, be it on, what line they use or a particular factor that they use in, growing up the cells, that actually render the cells not fit for purpose in terms of a clinical product at the end. So I would say that compared to established modalities, we live in the iPS cell space with a little bit more uncertainty, and we've learned an awful lot about the biology of the cells and controlling the cells over the last 20 years. And I think now things will start getting really interesting in terms of, clinical products and seeing a difference in patients' lives. And so a significant development that's happened in the iPS cell space just in the last few weeks is we've seen the first regulatory authorization of iPS cell-derived medicines in Japan. And that's basically a new medicine for heart repair after ischemic cardiomyopathy, and another medicine which is basically dopaminergic neurons that we have derived from iPSCs that would be used to treat Parkinson's, where that particular cell type is lost. So it's exciting times for the iPS cell field across the globe.
Well, that really is exciting, actually. Obviously, it's been like a long time coming of understanding the fundamentals, like you say, but now that they're actually managing to have an impact on patient, that's really cool. On manufacturing, then, so tell me a bit more about the differences between established modalities and iPSCs. Maybe someone with a traditional pharma background who's been working in established modalities. What do you think is the single thing they'd maybe find the most surprising about manufacturing an iPSC therapy?
Well, I suppose the biggest surprise is usually that the manufacturing process is actually inseparable from the biology. So in traditional pharma, you're producing a well-defined molecule. In iPS cell therapies, the product is a living cell population, and the manufacturing process actually guides the developmental trajectory of those cells. So, the process doesn't just produce the product, it determines what the product becomes. And that's why manufacturing in this field often feels closer to developmental biology and tissue engineering. So, there's a lot of experts in the field that come from, for example, blood transfusion medicine or bone marrow transplantation work than traditional pharmaceutical process development per se.
Did you have a sort of a moment in your career where you started to realise that manufacturing was the real challenge here, not just the fundamental science?
I remember, like, back in 2017, God, I'm so old, this particular point became very clear that, manufacturing was the real bottleneck, because I was working as program manager for the Global Alliance for iPS cell therapies, and we had just sent out, a rudimentary kind of questionnaire to all our members about how they define quality of their cells and how they assay for quality in their cells. And we got the results back, and the way our several hundred members were even defining what quality was were all over the place, and they were using completely different assays to measure different things. And it really pointed to the fact that unless there was greater alignment and standardization, the iPS cell field would be it would be unnecessarily retarded in terms of its progress. We had seen with other kind of cell therapy spaces that lack of standardization at a critical time in the development of the cell therapies meant that everybody was working off their own specifications and their own definitions. And of course, it's only global standards that are universally used that really give you the benefits of effective manufacturing. So I think during that time we decided to really start to learn the lessons from other ancillary areas of the cell therapy, and particularly where good progress had been made with blood transfusion work and bone marrow transplantation work, to bring a much more standardized infrastructure to the way people were developing iPS cell therapies. So that that since 2017, that's basically been on my to-do list since then. And so there's a number of different ways we do this. So as I say, the Global Alliance for iPS cell therapies help establish a provisional global standard for critical quality attributes for iPS cells that are to be used in a clinical context. And so we defined quality when it came to the stem cells. We then went on to start a series of quality round robins, where this is basically where we sent blinded materials to many sites worldwide and asked people to test for quality according to the way they normally do, and then give us their data back. We'd anonymize their data. They'd know what data was theirs, but we'd anonymize it and amalgamate it with everybody else's, so that they could see where their quality tested data agreed with everybody else's and where it didn't. Through doing those blinded round robin tests, we slowly built confidence around how we were testing for quality. I mean, this again will seem like completely alien and basic to, somebody who works in a CDMO around small molecules, but here we're dealing with, in some cases, release criteria that lack pharmacopoeial guidance presently. So, you can't wait for the regulator. You have to engage the regulator and work with all the other stakeholders in order to build the infrastructure that you need today. And that's kind of what we've been working on. I'm very excited about a paper coming out later in March where there's like the last 4 years worth of testing these blinded samples will be published, and it will basically show that people aren't aligned as much as they think they are in certain quality tests, particularly when it comes to biomarker expression analysis by flow cytometry. So I think I'm really looking forward to that being published and being out, because it's been a colossal amount of work done by the Center for iPSC Research and Application in Japan and the Global Alliance for iPSC Therapies.
So how do you go about defining quality attributes for living cells?
Well, ultimately you start off by getting as many different people in and listening to their perspective, because you're initially building a community, and it's a bit like you throw the net in the water, catch as many fish as possible, and then slowly drawing the net ever tighter, and the fish get closer and closer together. So you start off with probably quite a broad, open definition for what you think an iPSC line should look like if you're going to use it to develop therapeutic products. So there'll be obvious things like sterility, which we referenced earlier, alongside things like genomic stability, for example, or specific marker expression. So you start off with a broad definition, and then over time you slowly draw the net ever tighter, and the specifications would gradually go up and up over time. But what you'll find is that what I alluded to earlier, the horses don't gallop to the four corners of the paddock. You have the horses close together, so they're a better chance of understanding each other and reproducing each other's activity, so to speak. I'm using some analogies here that are, they can be quite useful, but sometimes in my Irish mouth they can be confusing. So stay with me.
Horses, fish. Let's see what other animals we can kind of use as metaphors for the rest of the podcast. Back to manufacturing strategy. How early does a biotech company need to be thinking about manufacturing when they're developing an iPSC therapy?
It's really as early as possible. The key element here is that early developers very frequently are forced to make decisions that really impact on the manufacturing side. So one of the reasons why I got involved in consultancy work is because you could see huge amount of resources being pumped into companies, and then the early developer who might have limited expertise in manufacturing making decisions that basically would make their therapeutic product unmanufacturable, or definitely not going to be getting approval from the regulators. So the idea that this specialized knowledge needed to be conveyed to early developers as soon as possible, so that we don't end up with just a large number of clinical trials. Instead, we end up with a lot of therapeutic products that have profound clinical impact on large numbers of patients. So, your question, when should a biotechnology be thinking about manufacturing? As early as possible. And with many pharmaceutical products, you can optimize the manufacturing later, but that's because the platform technologies there are well understood. But with iPSC therapies, manufacturing decisions actually shape the biological activity of the product. So you got to be thinking at it from very, very early on. And the way the cells are expanded, cultured, differentiated, all affect the behavior of the final therapeutic product. So that, it's unforgiving, but it means that you need to, measure twice and cut once, so to speak. So that the manufacturing is not just a downstream operational activity. Manufacturing is part of the product design.
I think that leads us on quite nicely, actually, to talk about the CDMO landscape. What does the CDMO landscape actually look like right now for iPSCs and for ATMPs in general? Is it quite mature supply base, or are most companies still building in-house? What does it look like?
5-10 years ago, the question would be, can you do it in-house, or do you have to do it with a CDMO? I think it's very apparent now that a lot of the technologies and the expertises are quite rare. So you do need to get in with a CDMO that has good personnel, with a lot of expertise in this particular area. So I think the CDMO ecosystem at the moment for advanced therapies and iPSL specifically is still developing. You've had kind of what I call the Chinese burn of, huge demand followed by reduced demand. So there's like a lot of CDMOs have gone through this hard business kind of position of initially they're scaling to meet huge demand, and then all of a sudden COVID and the demand is kind of reduced dramatically. So I think that the CDMO's opportunity now is in, I think, acquiring some of these other CDMOs and other individuals that have specialized knowledge in particular areas, so that we might be left with a smaller number of CDMOs, but they'll have a deeper expertise of how to get things done effectively for their clients.
Consolidating the sort of space through acquisitions to make sure that there's the right expertise there.
Yes, and I mean, I think one of the things I say to all my clients is that have a look around, see the CDMOs should at least be at the table when you're discussing standards, global standards. If your CDMO has no interest at all in global standards, that is probably a bit of a red flag, because we want to make things according to global standards so that they can be as cheap as possible, and as a result have the greatest clinical impact as we roll it out on across as many patients as possible. So CDMOs with a range of different proficiencies within them, like for example immune matching, an understanding of the importance of cell line selection. Like you'd be surprised that some CDMOs are kind of offering cells that are, on the face of it, they're not properly consented, and then in other areas they are deficient. So I think going after who works individual CDMOs and what success they've had in progressing things to not just clinical trial, but towards the market that looms ahead, I would say. So I think again, the space is quite small, so you actually know a lot of the people who are pioneers in this space. So getting them into the same companies so that they can have a synergistic effect for the client will be a definite opportunity for going forward. So I think we went through a period of having a lot of smaller CDMOs, and now, as you say, consolidation. And I think after this difficult time, the result will be CDMOs that have a stronger expertise in the area. And that can only be good for clients going forward.
So you've talked about a few, but what are the other questions that you think that you advise clients that they must be asking when going through CDMO selection?
Well, companies often focus on kind of like operational factors like capacity, cost, and timelines. And these things matter. But the more important questions are strategic, like does the CDMO understand stem cell biology? Do they understand the history of, maybe what's happened in other cell therapy areas? And do they understand the importance of global standards and, standardized manufacture? can they help define the product and the manufacturing process? Can the process scale beyond early clinical trials? There are still people with the mindset of, we'll get it into clinical trials and it'll be grand. And really that's, investors would not be happy with that, because, investors are not just interested in getting clinical trials. They obviously want something that, can be a big game changer in the market. So working with a CDMO that has an holistic vision for translation well beyond just clinical trials. So, how do we do things right first time so that we can roll it all the way through, from early stage development all the way through to commercialization? And if needed, can knowledge transfer elsewhere be supported? Because in this field, the CDMO often becomes a co-developer of the manufacturing process.
What's the most common mistake you see when companies try to scale iPSC manufacturing from lab to clinical grade?
I think they often, as I alluded to a few times, they make decisions they don't understand the consequences of. They often underestimate how difficult it is to define the product. With traditional drugs, the product is a molecule. With cell therapies, the product is a population of living cells. And, we've touched across a number of different areas, like, irradiation, sterility, and, you can't irradiate living cells because you kill them, and all these other kind of unique challenges that are particular to cell therapies and pluripotent stem cell therapies in particular. Now, there's an ongoing dialogue between the regulators and developers and other stakeholders in this space, because they want to understand identity, purity, potency, and genomic stability of the cells. So, there's a very strong dialogue between the regulators and the developers and the CDMOs, trying to work out how much is too much. We don't want to specify for things we're familiar with and under-specify for things that we don't know enough about. Just measuring what we like measuring, so to speak. So everything that we look at and everything that we certainly demand as mandatory, like release criteria, have to be very good predictors of patient risk. We shouldn't be measuring things just because we've traditionally measured them, because ultimately, in order to satisfy the regulator, we need to get a firm hold on what are the risk of these cells, if any, and measuring that risk when it comes to, developing these cells as a therapeutic.
What are the most sort of costly surprises when you're working with clients?
Oh, okay. I think the most insidious kind of problems that arise are the ones you don't really realize you have. You're sitting across from another stakeholder and they're agreeing with you and you think you're on the same page, and then it turns out that even though you're both using the same language, your understanding of the words that you use are different. So a classic example of this is when you're converting academic protocols directly into GMP manufacturing processes. So academic protocols are designed to demonstrate, biological mechanisms, but manufacturing processes need to be, they need to prioritize reproducibility, scalability, and process control. And those are very different design objectives. So even, little things like developing good, clear, actionable, SOPs, so that they would be the kind of the common mistakes, I think, when it comes to, getting into the GMP side of things. I mean, you'd be surprised at how many early developers think that GMP is somehow a denoter of quality, and it's not. GMP is essentially a documentation process. There's no such thing as GMP grade, and yet you'll see a lot of marketing teams go out and infer that their product, because it's GMP grade, is all early developer needs in order to get a therapeutic product all the way through. And it's it's very much a case by case kind of position. Like for example, a wise man once said to me, you can make a GMP bucket of muck, in that if you document it, and if you repeatedly get a bucket of muck at the end, that will fulfill the GMP side of things, because it's consistent and you're getting the same outcome each time, and it's well documented. But of course it says nothing about quality, and that like GMP is part of things, but quality and understanding what are the specifications we need here in our therapeutic product, that needs to be thought of very, very, very carefully.
Yeah, that's a really interesting point, actually. So if you could change one thing about how the industry approaches ATMP manufacturing today, what would it be?
Well, I think just, it's very important to paraphrase. So don't assume that people understand the same thing by what you understand. And like, I referenced these quality round robins recently, and I have a lot of experience dealing with not even different companies that are having trouble getting consistent outcomes across different sites or across other stakeholders. It's because they're not as aligned as they think they are. So you have to take your time and really go through the detail of what you're doing, paraphrasing what the other stakeholder is telling you, so that you understand them and they understand you properly. It's kind of a case of the more haste, the less speed. You really have to sit down and painstakingly develop an holistic vision for translation, and to really double check all the assumptions that you're making before you start a manufacturing run. So for example, if you're using one quality test and I'm using the same quality test, do we get the same result on the same material? And if not, then that assumption is going to really scupper things when it comes to trying to understand the manufacturing data.
Finally then, where do you see this space in 5 years?
In 5 years' time, the CDMOs will have deeper levels of expertise. I think there's going to be a continued rapid growth in this area, because it's a universal platform, and you could see how for example, a lot of pharmaceutical companies would really love if they could switch from taking primary cells from patients again and again to just having a small number of iPS cell lines that they could use as a manufacturing unit to develop a whole range of therapeutic products. You could see that once we have cells that are appropriate, then we could make a lot of different products from those cells. So I think that's we're going to see obviously more clinical trials. We're going to see more developments around, in particular, things like genomic stability and how we test for it, and functionality of the cells at scale. So we're developing all these analytics, because they're quite limited presently, but they're essential. And once we get those analytics in place, then we'll be able to not only scale the cells, but verify that the cells will behave in a manner that we expect once introduced into the patient. The other thing is AI. AI will probably help in a number of different areas. I think one area that it'll really help is in teaching the early developer to have a more holistic vision of translation. So, at the moment, or, in the times gone by, the early developer, would struggle a lot with their initial engagement with the regulator, because the regulator uses language in a different way sometimes to what the early developer would use. And I think what AI will help with is they will allow you can use the training guidances from the FDA and the other regulators to train agentic AI, and then you can come along as an early developer and ask loads of questions to the regulator AI, and that will allow you kind of get a dress rehearsal so that when you do come to meet the regulator, you'll be far better able to ask their questions in a meaningful way and give them the context they need to give you actionable answers. Does that make sense?
Yeah, sort of like a translator tool from development to regulation, how to sort of bridge that gap.
Yes, and also, you come in with a deep understanding of your therapeutic product, and the things like classification of your therapeutic product, and I think AI, based on the guidances that the regulators produce, will make teaching early developers far easier. That sometimes you needed to know what was the right question to ask, but I think with these AI interfaces, they will help the early developer understand better what the regulatory responsibilities are, and also what their own responsibilities are, because I think it's quite common mistake I see that an early developer thinks it's all on the regulator and the regulator will tell me what to do. And yes, that's true if it's an established modality, but if you're in an emerging modality, it's your job to engage the regulator as much as it is the regulator to engage you. And you have to learn how to effectively engage the regulator. So you got to learn, why is the regulator there? What do they mean when they say this and that? And then you have to supply them with the context so that they can give you answers that you can then action. But, without a context, like, for example, the way I describe it is, going into a court of law and asking an attorney, give me a checklist to prove innocence. And it's like the attorney is going to turn around to you and say, well, that depends on what the crime is and, all these other things. So unless you give me a context of, what the accused is being accused of, I can't give you a straight answer. And I think a lot of early developers fall into that trap because they go to the regulator and the regulator is kind of giving them non-actionable answers, and they think that the regulator is being obtuse when actually it's just the regulator is waiting for content-specific information to frame their answers.
Cool. Stephen, thanks a lot for sharing your advice and your thoughts on here.
My pleasure, Dora.